Hello, my name is Jack Doering, and I did my post-over PCR's use on solving crimes with DNA evidence. PCR is short for polymerase chain reaction. It is a method used to prepare specific DNA sequences for copying. We were tasked with using DNA from 3 suspects and the murderer's DNA to determine who committed the crime being a murder committed in a house. We hypothesized that through the use of the PCR method, we would find a match between one of the suspects and the murderer. For this experiment, we mixed the purified DNA samples with nucleotides, primers, and polymerase. Then we placed the mixture into the PCR machine. After the PCR process was completed, we placed the products into an electrophoresis machine. Lastly, we analyzed the DNA bands shown by the electrophoresis machine for similarities. Figure 1 shows the DNA bands from the electrophoresis machine, and on the right, Figure 2 shows the percent similarity in DNA bands between the murderer and the suspects. Using this, we were able to determine that the rival scientist shared a 100% similarity with the murderer's DNA, while the postdoc and PhD student did not. This led us to the conclusion that the rival scientist was the murderer. The use of the PCR machine in this experiment suggests that the PCR process can be used to determine DNA similarity and its possible use in crime where DNA evidence is present. During the experiment, we were not told the exact measurements for the primers, nucleotides, and polymerase. Also, some of the suspects' PCR products were made without our supervision. We also assumed that the machines and lab were maintained correctly and cleaned. This concludes my presentation.